We're going to end the first day of the neuromuscular system by reviewing membrane potential. And I actually know that we talked about this on during the first week of class, but I want to reiterate this and talk about it in a little bit more detail than I think I did on that first one. And the reason why membrane potential is so important, and here are the objectives that we've got here. So what is membrane potential? What contributes to resting potential? And why do we care about the pump? Now, the reason that it's really important to understand membrane potential is because anytime you do anything, a neuron sends a signal, correct? And not even just a neuron, but your muscle cells also have potential. And remember, we're talking about electrical charge, and so things have to change with that membrane potential in order for anything to happen. Okay, if there wasn't a difference in charge between the inside and the outside, then you would just be nothing. Okay, and so I don't know how better to say that, but just a reminder: ions and other things like proteins have a charge, and so membrane potential is the difference in the charge of voltage between the inside and the outside. Okay, so we're basically taking the charge of the inside and we're subtracting the charge from the outside. All right, so for example, if we have plus 5 minus 10, 5 minus 10 equals plus 15. Okay, this can get a little tricky at times because so many of us don't think about subtracting and adding negative numbers, especially if both numbers are negative. I think we get a little tricky with that, or it gets a little tricky for us. And so if you're not sure, use it. Like I would legit say, use a calculator. Okay. But it's pretty straightforward. So inside minus outside, that will give us our membrane potential. Okay, and so here we've got our cell membrane, and we've got the sodium-potassium ATPase pump. And so there are 4 things that contribute to membrane potential. And these slides are right from 220, so I'm not going to just even pretend that they're not. They are. And so we've got concentrations of sodium and potassium. So we've got much more sodium on the outside than the inside, and much more potassium on the inside compared to the outside. Now, if we just take the amounts and concentrations of both of those, it really doesn't change membrane potential, okay? If we added all of them up and we just focused on sodium and potassium, there would essentially be no difference in charge, okay? But what happens is we've got a pump. And so this pump, the sodium-potassium ATPase pump, every time it goes, we take 3 sodium out. Okay, so we put 3 out here. And then we bring 2 back in. Okay, what that essentially does is that results in a negative charge. Okay, so that makes it so we're more negative by 1 every time a pump runs. Okay, and we do this a bunch of times, and eventually, we're going to get, and I'm sorry for this animation or the way this works. I thought I had fixed this, but this is a number you'll never need to know, but I'm just giving an example. Then we might end up with negative 10. Okay? So there's also a bunch of proteins and other things inside the cell that are negatively charged, and those are really important, especially as it relates to the action potential, which we'll talk about next on the next video. All right, and so proteins, metabolites, acids, they're negatively charged. And so we're just going to add a couple of negative charges there. And so that brings us up to minus 20. Okay, then we've got these things called leak channels. And remember, leak channels are hallways. There's no door. There's no opening. It's an opening in a wall. Okay, and you can't close them. Okay, and so what happens is ions go down their gradient, so flow down gradient. So potassium, more concentrated on the inside, goes outside of the cell. Okay, and every time it does that, it's like we add a negative charge. So we do that a bunch of times, and we've got a negative charge. All right, and so maybe that makes our membrane potential negative 80. It's important to remember, though, basically as soon as this potassium goes out, it's pumped back into the cell through the sodium-potassium ATPase pump. Okay. Okay, and so that's 3 things. And so notice so far, the pump, more negative. Proteins, more negative. Potassium leak channels, more negative. All right, so the last thing we've got is we've got sodium. Okay, and so we've got a sodium leak channel just like we've got a potassium leak channel. All right, and so what happens is sodium flows down its gradient into the inside of the cell. So every time that happens, it's like we put a negative charge on the outside, and so that negative 80 becomes negative 70. Now this negative 70 is the only number that you need to remember. Okay, this is resting potential on a neuron. All right, but just like with potassium, when sodium comes in, it's almost immediately pumped back out. All right, now this negative 70 is an equilibrium point. Based on all the doors that are open, the ion concentrations, the pump, this is equilibrium. Now, some of you, when you hear that, are going to think that nothing's moving. No, everything's moving. Okay, potassium's constantly going out, but then being pumped back in. Sodium's constantly going in, but being pumped back out. Okay, and so right now we're at an equilibrium point, this resting potential of negative 70. The only way that we'll get away from this negative 70 is if something changes the system. And that's where an action potential comes in, and we'll talk about that next time. All right, but I want to focus on this the sodium-potassium pump. Okay, now just a reminder, it requires energy because this is active transport. Okay, we're going against the gradient here. We've got a lot more sodium outside than inside, but we're pumping that sodium from the inside to the outside of the cell, going against the gradient. So it's pulling sodium out, and it's pushing or putting, or however you want to put it, potassium back in. And what this does is this helps maintain the ion concentrations between, or the gradients for both of these ions. Okay, this produces the negative membrane potential that we have in neurons. So what I want you to do for a second is think about, well, what happens if we kept everything, just kept the proteins, kept the leak channels? What would happen if we didn't have the pump? Okay, and so here we go. There wouldn't be a gradient between the inside and the outside of the cell. Sodium would continue to go in until it's balanced. Potassium would continue to go out until it's balanced. Now, it wouldn't be exactly like that. It would be really, really close, though. There might be a slight bit more of positive charge inside, but it wouldn't be much. Okay, and the reason we might have a little bit more positive charge is because we want to balance out those proteins. But it would be a small gradient. Okay, and so what would happen is we would essentially eliminate any ion movement when we open additional channels. Okay, remember, we're at an equilibrium point with the pump, with the leak channels, with the proteins. But if we change the system by opening doors, which is what happens when an action potential starts, we need that gradient so that sodium can go in or potassium can go out. If there's no gradient, it doesn't matter if I open a door, nothing's going to move. Or if it's a small gradient, things are going to move very slowly, and that's not great either. And so really, without the pump, there wouldn't be any action potentials. I believe I mentioned this on week 1, but it's important to remind you, or I want to remind you, that they estimate something like 40 to 50% of the energy used by your brain maintains or runs this pump. And so it's super important. And so that's where we're going to end the first day of the first week of neuromuscular.